Okay, hello everybody. Today I want to talk about analyzing proteins, particular protein structures. And maybe before I do that, I'm going to show you a piece of software and another website, very similar to GenBank, called Protein Databank. This is where our structural database lives. And then we're going to be using a program that's similar to Mega, that's known as UCF Chimera, which again allows you to sort of access that database directly once you've installed the software. So these two places where you can obtain this information, you can download structures from the Protein Databank, and then you can also open them and explore them and visualize them using this molecular modeling software from the University of California, San Francisco, known as Chimera. There's a cross-platform new version known as Chimera X that has been under some development, and you might want to check that out. It's probably only about half to three-quarters of the way there. It's intended to replace Chimera in the next couple of years. It's very cool. There's a link here to it. It's very, very nice because it actually has a lot better shading for visualization. And it also has the ability to use virtual reality headsets and things like that. So it's a lot of fun. But that's still a work under progress. Now, before we talk about proteins, I want to talk sort of about what proteins do. Proteins are, we'll talk a lot more about this in class, or they're kind of those nano machines. They're the machinery that's created by the genes in our genome. And these interact with each other in a very complex way. There's no gene that sort of works in isolation. There's no protein that works in isolation. They work in very complicated large networks of interactions. And when we develop pharmaceuticals and drugs, we're often looking to interfere with those pathways, what we call biochemical pathways. We either want to activate pathways that are normally not activated, or we want to turn off pathways that are active when they shouldn't be, by blocking the natural binding sites of proteins with particular drugs. Now, there's a website called KEG, short for Kyoto Encyclopedia of Genes and Genomes, but it's really a wonderful place to kind of explore the pathway, the underlying networks of things. It's called a knowledge base. It combines genes and orthology and genomes with information about the underlying networks and pathways that they play into, and the diseases related to those pathways, to deficiencies in those pathways, and the drugs that are used to target that. So just to show you the power of this, in the class you're going to have a project at the end where I'm going to ask you to explore a particular drug of interest and how it works on a protein system in the cell. If you go to KEG drug database, you can kind of do a search here. And I'll show you a couple of things here. Let's look at OxyContin. This is the drug that's been blamed for the opioid crisis. If we do a search here, we can quickly find the name of the drug. The actual, the brand was OxyContin, but the oxycodone hydrochloride is the name of the drug. If you're using a generic form of the drug, it's actually usually called this. There's entries that you can link to from here. So the entry for this drug is here. Gives you all the information about it, who makes it, when, gives you a structure, molecular weight. You can also go back here, and there's a section here for target pathways. So if I follow this link, it says here neuroactive ligand receptor interaction. So this is a particular place in our bodies where that's targeting. You can see here now these lines are actually representative of cell membranes, and these boxes on those lines are membrane-bound protein receptors. And what's cool about this is there's a whole list of them here. These are all the neuroactive ligand receptor interactions, and this OPR gene here is in red, and that indicates that this OxyContin is actually targeting this particular one out of this whole page here. And you can click on that and you can actually follow it to It's information about this particular gene, the name of the gene, all of the names that it may use, opioid receptor delta. I can learn more about other drugs that target it, so I can go back to different kinds of drugs. So this is really kind of a rabbit hole. You can really go back and forth and learn quite a lot. You can learn about diseases related to it. So here we have eating disorders. If I follow this to the disease, I can find a description of the disease related to that particular gene. Now, I'm not really, talking about opioid overdoses anymore, but I can wander around in this database and I can sort of surmise there might be a link, say, between these eating disorders and perhaps the way the opioids actually work, because they're related to the same underlying pathway or gene network. Let's go back back to the beginning here. I'll show you another example. Sorafenib. This is a drug that targets a particular kinase protein that is important in pathways that are activated in cancer. So here's the drug entry. It's also branded as Nexavar. Here's the drug. I can go back to the target pathways and I can see, well, there's a lot of target pathways here. And the MAPK signaling pathway is the one that's often involved in cancer. And here I can see where that drug actually, targets. RAF1 or BRAF are both targets of this drug. And I can follow that to a page here describing the BRAF proto-oncogene. It's a serine threonine kinase. So you can find out quite a lot about proteins and drug interactions just by wandering around in here. This diagram shows you the full MAPK signaling pathway and how it triggers and affects essentially cell cycle. So this is a pathway that's turned on and causes growth and proliferation. It's important when you're a little embryo that you grow and proliferate your cells. It's not so important when you're an older person, and we often call that cancer. So this pathway is problematic when it becomes turned on, and a lot of drugs are targeted to sort of interact and turn this off. We'll talk a lot more about cell signaling in the course. You notice probably that these arrows indicate signaling events. Positive arrows are generally activating, and then when you see these little blocks here, those are these little feet, those are repressive sorts of signaling. So you can see repressive here. You can also see plus P's and minus P's. These are indicating what we call phosphorylation events. A lot of times signaling of genes can just be the presence of the gene itself, in which case you've activated it and turned it on and up what's called upregulation of the gene. You're just making more of it, and that protein has an effect. But a lot of times proteins are activated or inactivated very quickly. They're present, they're floating around in the cell, but they might be in an inactive state because they're not phosphorylated. And that's simply the addition of a phosphate group. There's a lot of enzymes that move these phosphate groups around and put them on and take them off again. And those are that's actually what kinases do. We'll talk more about this in the class, but you can see there's a lot of action here going on with phosphorylative signaling. So anyway, this is a great database. You're going to be using it pretty extensively to research your own questions later in the course. And that's the KEGG knowledge base. So back to talking about analyzing proteins and protein structures. So you're going to want to install Chimera, and I'll show you that in a minute. Protein Databank, this is where we go to find our structures. So for instance, if I wanted to look up my BRAF and sorafenib, I could do a keyword search here and look for a structure of that. Is there a structure of that drug bound to that? I actually already know the PDB ID for that, so I don't necessarily need to do a search, but I can pull it up here. All protein databank entries have a short, what's called a PDB ID. It's usually a number followed by 3 other characters. These can be letters or numbers, these remaining 3 characters. It's never more than 4 characters long. Now if I search this, I have a complex of wild type or essentially human BRAF and this drug called sorafenib. And I can download that structure. I can follow that to a page here. I can actually look at it in 3 dimensions on this if I want. And I can download the PDB file in PDB format and save it and then open it in Chimera. Or I can actually just fetch it from Chimera as well. So, so let's talk a little bit about that in a minute. Another thing I want to point out about Protein Databank is the link to Learn, or PDB 101. There's a really great article here called Molecule of the Month, and you can learn a lot about the way different molecules work. If we were to go to, I think, coronavirus, for instance, there's a lot of resources related to coronavirus, and I could read them here. There was a July molecule of the month, a June molecule of the month was about the coronaviruses. Now, the coronavirus has a very large spike protein, and it was discovered through a method. It's a very large protein, so it had to be discovered using a method called cryo-EM. Generally, there's 3 different types of methods that generate the PDB files, or the protein databank files that you find here. The bread and butter method is and always has been X-ray crystallography. That's where we try to crystallize the program. We take it to a high radiation light source and get a diffraction pattern from it that lets us predict where the atoms actually are. We also have solution NMR. This is generally very small proteins. If you open up a file and you see that it has lots of backbones that are nearly overlapping each other and it's relatively small, that's probably an indication it's an NMR solution structure. These are nice because they're limited in that they can only look at small structures, but they're nice because they show you sort of the conformation in solution rather than in a crystal. But there's a new method coming up called cryo-EM, where it's actually frozen in a glassy ice, and then computers are used to reconstruct the structures from many large patterns that are seen by the protein sample frozen in its frozen form, and that's called cryo-EM. Cryo, or very cold electron microscopy, is what that stands for. A cryo-EM structure that was recently solved was this actual spike protein here. If I can remember what that is, I think it's 6VSB. Let's try that. Yeah, here it is. So 6VSB. This is the structure of the spike protein that's related to COVID-19. So we're learning about how this spike protein enables the virus to attack our cells is a major area of research right now. In my own lab, we've been looking at this as well, studying some of the dynamics related to it. Let's go ahead and take a look at it using the Chimera software. So if I actually go into Chimera, I'll get a page like this, and I can I'll register that later. I can use the fetch command down here. I can open up the web page that you just saw by clicking this web page link and see how I'm back there again. I can also just fetch the ID if I already know it. So if I did 6VSB, now you see the big spike protein. So these are generally shown in what's known as ribbon format, and that shows us the important protein secondary structures. The beta sheets, which are these flat arrows, the alpha helices, which are these spring-like coils, and then just these cords that are kind of running around are known as loop structures. So right away you can see the loop structures. If I go to my ribbon preset, I can actually color these. So these are actually 3 different chains of protein that are assembled together into this spike protein nano machine. Now, what's important about this and how it works is this little guy here. This is an open conformation that was recently discovered as it fuses into the human cell. And what it appears just by looking at the structures, it looks like it opens down here, and then it allows this structure to sort of flip out. And so when this, you can see these three matching domains here, here, and then here. This one flips out, and then it can interact directly with what's known as the ACE2 receptor on our cells. If we go back and I close this, I can show you another structure which shows that interaction. And so if we were to fetch another structure known as 6M17, we can see that domain interacting with its receptor. So this was the domain on the end of the spike protein, this little red guy here, and you can see it's interacting with a very large protein that sticks out. This is a membrane-bound form of the protein. This is when you open up things in Protein Databank, you often see mirrored images. So you can see that this bottom structure is a mirror image of this top structure. That's very common if your method of developing is X-ray crystallography, is to see these mirror images. And typically what you would do is you would delete it. So if I Control Alt and then pull a swipe here, and I can actually select, I can up arrow until I select all three of those chains. And if I go under Favorite, Command Line, and I click on, and I write Delete Selected, I can actually delete that. And now I'm looking at the actual protein I'm interested in, so without that mirror reflection. So here's the viral protein, and this is the human ACE2 receptor. This part here sits in the membrane of our cells, of our lung cells, and this part sticks out, and the virus recognizes this when it sticks that huge spike out. Now, just to show you one of the things you could do, you might ask yourself, well, how does I want to see the whole spike sort of interacting with this. So I could actually do a structural alignment of this portion of the spike protein with the other portion. So if I were to go back and say I want to fetch 6VSB, now I've opened up that spike and I have both of these. There's a tool here that's really very useful called Structural Comparison and Match Maker. So I could actually match my two structures by running this. It might run a little bit on the slow side because these structures are so large. But we'll kind of let that run and come back and sort of check it out in a minute. In the meantime, let's go back to sort of Protein Databank and sort of show you a couple of other things that are very cool about it. So on the Protein Databank, if you were, there's usually links to the publications in which the structures were published. And you can search this. This takes you back to NCBI, which was PubMed, which was another one of those databases in the NCBI. And it actually gives you links to papers, and you can actually often find full-length you can often find links to the full-length papers. You can read about them here. So you could read all about coronavirus and how it works in this paper here. The other thing you can do in PubMed is you can do author searches. So, my last name and my two initials. You typically would put last name and then the two initials of the author to do a search here. Now you can see my you can see at least some of my publications here for my list here. So so that's an example of PubMed. Let's go back and take a look and see if we've got a structure. I'm going to go ahead and pause, and I'll restart the video once this is completed. Okay, that took a few minutes, but it looks like it's finished. We can see now that what this has done is it's actually overlapped that little finger of the spike protein that sticks out here to interact with ACE2 with the same structure that was on ACE2. And if we were to sort of zoom in and look at that, you can see how similar those two structures are and how well they're overlapped. So this can be sort of a nice way to align structures if you need to, or to explore larger interactions such as this. So there you have it. This is the spike, the coronavirus spike protein actually attacking the human receptor on the cell and showing how they would actually interact in a recognition event. Let me go back to a simpler example, which was that sorafenib drug bound to the BRAF kinase. So that was the target of that cancer, that inhibitor drug. So the PDB ID for that is 1UWH. And you can see right here, again, I have sort of a mirrored image with it. If I go and look at my ribbon presets, and I can ignore the red structure and sort of focus more on the blue, I can actually now see, let's go ahead and just, again, delete that. So again, under favorites, command line, delete selected. And so here's my my BRAF protein, my BRAF kinase protein, and in a site where a chemical signal known as ATP would normally interact with this protein, there's a drug structure here called sorafenib. So if we were to zoom in on that, you can now see that that drug sitting in that ATP binding pocket. These little parts that are sticking off of the chain here, these are actually indications of the amino acids that are interacting with the drug and actually holding it there. So a couple other things you can see here that we want to talk about. There's this dotted line here. You can see that there looks like there's a part of the loop that's chopped off, and it's only connected by the dotted line. This happens whenever in X-ray crystallography, loops are so dynamic and moving, they don't often crystallize very well. So we often miss that from the structure. There's a tool, it takes a while to run, so I'm not going to run it in here, but if you go under structure editing and you go to refine loops, you can actually open up a tool that's called Modeler, and it will use the protein data bank. You can read about that method here. It uses the protein data bank as a comparative modeling data set, and it will rebuild the loop for you based on all of the different kinds of kinase structures that are already present in that database. It's something called homology modeling. It's very powerful. Won't run that here, but that's an example. It would be very important to refine this loop if I was going to be doing any sort of analysis of it. I can also look at this structure in other ways as well. So another way that I can go back here is I can go to the all atom preset, and now I can look at all of the atoms present. Now, again shown as a wireframe. Now, X-ray crystallographic structures are missing all of the hydrogens. This is because hydrogens don't often or are unable to really diffract X-rays. So there's a tool in here that you can use. Again, under structure editing, you can say add H. I can go back and add all my hydrogens back to this structure. And now you can see, now I have a much more complete structure. Now, yeah, I'm not going to really see hydrogens in that ribbon preset, but if I go back to my ribbon preset, since I have only one chain left in my structure, it'll actually color it as a rainbow. So I can look at essentially this is the C terminal or the end in red, and then in blue I have the N terminal. So the protein starts here, and then the chain goes and sort of follows the rainbow until it ends up over here. So that's another way you can look at proteins. I can also look at surfaces as well. So I could say I want to show a surface. I can put a surface onto that, and this would show you sort of, this is essentially what we call an electron density map. It sort of shows you where the electrons would be. I can also go back to the surface and add a level of transparency to it. So if I wanted to sort of see more of what's happening in there, I can go and add a high level of transparency to it to varying degrees. So that can be fairly useful as well. Another preset I can use is showing the hydrophobicity of the surface. So if I go back and this is essentially showing how water-loving or water-fearing the atoms are in the protein itself. So if I go back and let's turn my transparency back up now. The blue indicates hydrophilic and the red sort of hydrophobic regions, and you can see that the binding site is sort of generally hydrophobic. So, so there's a nice another way to analyze structures, if you will. There's lots of tools in Chimera for aligning things, for analyzing structures, and doing molecular dynamics, even doing molecular simulations or simulations of motions. You can create movies of motions. You can do a whole lot of things, and we'll talk more about sort of what you can do with Chimera later on in the class. But I wanted to give this as just a sort of a general, quick introduction to some of the software we'll be using in the class. So again, just to wrap up, Protein Databank is where you find these structural databases. And again, this is the BRAF that I just showed you. This is a structure I could download, I can open it, or I can fetch it from the Protein Databank. And then I can analyze it in the software called UCF Chimera. Okay, I'll conclude it there for today, and thank you very much.